Thank you very much for the introduction. So I'm happy to present you Tiamat, a tiled image access manipulation and analysis toolkit for visualization and analysis of large scientific image data sets, which is basically a software that we developed to visualize a whole variety of brain image data. As you can see here, we deal with cell-stained images, we deal with cell segmentation, transmittance images, fiber orientation maps, block phase images, Z-stacks, and also 3D reconstructions of them. And what all these data have in common, they are huge. So we are going from 15 gigabyte for a flat scan of cell-stained images to 300 gigabyte for PLI or Z-stacks. And if we think of reconstructing whole brains, we need to deal with up to 8,000 sections, which easily boosts the amount of data that we need to handle to the terabyte to petabyte range. And this already shows us a problem that we face, because usually on computing infrastructure, we don't have this size of memory. So we are dealing with memory limitations when holding these whole slide scans for analysis or processing. Which basically means that we can always only access parts of the data at a time for visualization or analysis. Another aspect is that we have, if we want to create 3D brain models, we usually have to apply transformations to the data, like deformation fields, affine transformation, which always mean that we need to restore the transformed result somewhere. So we are basically creating, each time we create a 3D model, we are creating a copy of our 2D images in this new image space, which duplicates the data. And if we think of the terabyte to petabyte range, that's really duplication of huge amounts of data. Furthermore, if we want to perform analysis, we have a whole bunch of tools and file formats that we can choose from, and each tool basically comes with special needs for file formats. So each of them has specific requirements on the file format, and we often find ourselves to transform between file formats for each analysis tool and perform data conversion. So our suggestion here is to use the software that we develop, which is Tiamat, which basically supports multiple image formats. So at the moment it's BigTIFF, HDF5, ZAR, but it's so designed that each time there's a new data format or some specific requirement, we can just extend the list of supported data formats, because it's like a framework that we develop that can handle different types of formats. And instead of transforming the whole data at once, we just transform like the current viewport that's of interest for the users, so we can apply transformations on the fly. So we just transform the data when it is requested by the user, for example, to perform tiling, color maps, spatial transformation, or also creating image stacks. And third, we want to support many tools. So at the moment we have Napari, NeuroGlance, or OpenSeaDragon interfaces, and each of them are replaceable with each other. So we can use many tools with many file formats and apply different kind of transformations to the data. So how does it work? We basically want to connect the user with the data by this kind of pipeline. Where we first read the image and metadata, perform transformations on it, like spatial transformations or metadata transformations. And third, we want to expose the transformed data to the user, who uses interactive applications, browser viewers, or programmatic access to access the data. And how it works is usually that oh, it doesn't look pretty, but the user usually creates a request by coordinate slices he's interested in, like the region of interest he uses for his analysis. And this is then transformed with Tiamat into a bounding box, which we call the accessor, which basically defines the range of pixels that need to be requested from the image to serve the request of the user. And since we are also dealing with transformations, That's not always the plain request that we need to load from the image, but if you, for example, think of rotations, we have to expand the bounding box of the requested data to avoid cropping or padding effects in the data that we transform. This is then looked up in the original image, then read as an image chunk, transformed, then stored in an image result, which is then used by different viewers that we provide interfaces to allow access for the user. So after this toy example, let's have a look at some brain imaging data, for example, this 1 micron image, where we already saw in few talks today that we can perform cell segmentations on them. And with Tiamat, we are now able to do this in real time. So the idea is that we first use Tiamat to use the tiling strategy to provide it to Neuroglancer, to first just have access to the original image. And then in a second branch, which is a second service instance of Tiamat, we apply the same tiling strategy, but this time we apply the contour proposal network that was developed by Erik Abschult in our lab to create a cell segmentation out of the image, which is then also served as a Neuroglancer interface and then can be used to overlay the 1 micron cell-stained image with the cell segmentations. And this is already integrated in a development version of Zebra Explorer. So if we have a look here, we can have a look at the whole 1 micron scan. And now we are able to click on this contour proposal network. And now for the requested view of the user, it's computing these cell segmentations in real time. So they're not stored somewhere, they are not like duplicated data, they are just computed by the AI model the time the user accesses the specific viewport here. And we cannot only apply it to single plane images, but we can also apply it to 3D reconstruction of brains. For example, here we have block face images of a whole hemisphere stored together with affine matrices that can then be used to create a 3D model of the brain. And instead of like duplicating the data, transforming it, we see here on the right a just-in-time computation of the transformed volume. So it's not stored on disk, but it's just computed as the user accesses the data. And here, when we zoom in, we see that it's loading like higher resolutions on the fly. So the initial volume is calculated on a coarse resolution so that we have access to the whole volume. And if we zoom in, it's loading the higher resolutions on the fly. And we can also navigate the original high-resolution block face image in the viewer here. And as a last use case, if we think of 1 micron reconstruction of a whole brain, we easily beat the 1 petabyte range. And we cannot, like in the development process of the 3D reconstruction, each time we have a optimized reconstruction, recreate the whole volume, store it somewhere for visualization. So here the idea is to use the original scanned data, provide it with the deformation fields, and also perform the 3D reconstruction just in time. So here you can see it for a slab of BigBrain 1 micron sections. And here you see that it's loading the higher resolution now just as the user requested. So there are deformation fields applied, Z-stacks that are loaded. So it's taking a bit of time, but you see it's working. So of course it would be faster if we just copy the data, but with this approach we don't need to create a copy each time we want to access the data anymore. Yeah, and then at the highest resolution we can also navigate then through the volume, and it's loading the data that's requested. So in conclusion, what we achieved with Tiamat. We decoupled data access from visualization and analysis workflows, which makes us quite flexible with the file formats or tools we want to use, because each time we want to use a new tool or a new file format, we can easily extend Tiamat because it's like plugin based. And if you want to check out our GitLab project, we are also happy for suggestions or pull requests for your specific data or your specific toolkits to extend like the application of Tiamat. And third, we can now avoid data duplication, especially in the development process of 3D brain models, by performing these deformation and transformations in real time. And by this, I want to thank all the collaborators, the Big Data Analytics Group led by Timo Dickscheid, and the Institute of Neuroscience and Medicine, INM1. Thank you.